In a remarkable display of academic excellence and engagement, 5 students from Illinois Wesleyan University IWU made a significant impact at the recent National Science to Policy Network NSPN Symposium held in Seattle. This event attracted a diverse group of participants from across the nation, but IWU stood out as the largest single cohort of student researchers selected to present their work. Among the 5 attendees, 3 students, Habiba Hamitova, Amina Quadri and Paige Griffin, had the honor of sharing their research findings, collectively representing a quarter of all student presenters at the conference. The trip, organized by IWU's Science to Policy Network registered student organization and led by Joelle McMillan, provided attendees with invaluable opportunities to network and engage with leaders in technology, science and policy. The discussions revolved around pressing global challenges including climate change, public health and privacy, underscoring the importance of interdisciplinary collaboration in addressing these issues. Amina Quadri, a data science major, expressed pride in her university's commitment to student research and its encouragement of participation in wider conversations. "It made me proud to be part of a university that values student research and encourages us to participate in conversations that matter on a larger scale," Quadri remarked. Her presentation focused on the intersection of public health and climate change policy, specifically how climate change worsens health issues like heat-related illnesses and the spread of vector-borne diseases. Quadri emphasized the need for climate-resilient healthcare systems and equitable intervention, showcasing how academic pursuits can lead to meaningful societal change. Paige Griffin, who was majoring in both data science and environmental studies, presented her research on soil erosion and its implications for sustainable agriculture and dust storm prevention. Her presentation, titled "The Erosion Crisis: Incentives for Sustainable Agriculture and Dust Storm Prevention," highlighted the urgent need for effective practices to combat soil erosion, a growing threat in Illinois. Griffin shared that hearing from local politicians at the conference, including Washington state senators, reinforced the vital connection between science and policy. "This experience has lit a new fire within me," she stated, expressing her motivation to continue her research in light of recent environmental events. The spectrum of research presented at the symposium was further enriched by Hallie Smith, a behavioral neuroscience student who reflected on the potential for student researchers to influence policy changes. "We are likely less stuck than we believe we are when trying to make change and a difference," Smith noted, encouraging her peers to harness their passions for societal improvement. The engagement of IWU students at the NSPN Symposium not only highlights the university's commitment to fostering research and policy dialogue among its students, but also exemplifies the increasing role of young scholars in shaping public discourse on critical global challenges. As these students return to campus, their experiences are poised to inspire further involvement in the intersection of science and policy, paving the way for future initiatives that promote research as a tool for societal advancement. The impact of their contributions at the symposium is likely to resonate well beyond the event, influencing both their academic journeys and the communities they aim to serve. In a groundbreaking development in the field of gene therapy, a newborn baby named KJ Muldoon has become the first recipient of a personalized gene therapy designed specifically for his genetic disorder. This achievement, reported by a team of researchers from the Children's Hospital of Philadelphia (CHOP) and the University of Pennsylvania, signifies a monumental step toward treating genetic diseases that have long been deemed untreatable. KJ was born with a rare condition that prevents his liver from effectively converting ammonia, a byproduct of protein breakdown, into urea, which is then excreted in urine. This deficiency, caused by mutations in both copies of the CPS1 gene, can lead to dangerously high ammonia levels in the blood, resulting in severe neurological damage or death. Traditional treatments for this condition include restrictive diets and medications, but these measures are not always sufficient, especially for infants like KJ who are too small for a liver transplant. The medical team quickly recognized the urgency of KJ's situation. Cardiologist Kiran Musunuru and pediatrician Rebecca Aarons-Nicholas, along with a coalition of scientists, rapidly developed a personalized gene therapy. The U.S. Food and Drug Administration (FDA) expedited their application, allowing treatment to commence within a week of submission. KJ has since received 3 infusions of a CRISPR-based gene editing therapy, which has already resulted in a significant improvement in his condition. He can now tolerate more protein and requires less medication to manage ammonia levels. The therapy utilizes a sophisticated tool known as a base editor, which modifies the DNA at the site of the mutation by replacing an incorrect base with the correct one. This is achieved through messenger RNA mRNA that carries the instructions for creating the base editor. Encapsulated in lipid nanoparticles similar to those used in COVID-19 mRNA vaccines. This targeted approach not only corrects the genetic defect but also minimizes the risk of unintended changes to the genome. This case is especially notable not only for its success but also for its implications for the future of gene therapy. The personalized nature of KJ's treatment, developed specifically for his genetic mutation, contrasts sharply with traditional approaches where therapies are designed in advance for a broader set of patients. This shift toward individualized treatment could open doors for many patients suffering from rare genetic disorders. However, while KJ's case is a triumph, it also highlights the considerable challenges that lie ahead before similar therapies can become widely available. Factors such as regulatory hurdles, high costs, and a lack of widespread expertise in gene therapy development could impede the scalability of these innovations. Experts suggest that broader regulatory frameworks may be necessary to allow for the development of more generalized therapeutic platforms that can be adapted for different mutations rather than requiring individual approval for each specific case. This would facilitate a more efficient process for delivering personalized therapies to patients who urgently need them. Moreover, as gene therapies often come with a hefty price tag, ensuring insurance coverage and funding for research will be critical to making these treatments accessible. The financial landscape for gene therapy is fraught with uncertainty as companies may struggle to secure the necessary resources to bring their innovations to market. As researchers continue to investigate the potential of gene editing technologies, the excitement surrounding KJ's case serves as a powerful reminder of the possibilities within the realm of personalized medicine. The hope is that his story will not only pave the way for innovative treatments for genetic disorders but also inspire continued advancements in the field, ultimately contributing to a future where gene therapies can provide effective life-saving solutions for those affected by genetic conditions. In an inspiring showcase of youthful innovation and scientific inquiry, 2 seniors from the South Carolina Governor's School for Science and Mathematics (GSSM) have garnered prestigious recognition for their groundbreaking cancer research project. Audrey Howard from Anderson and Grace Zhang from Simpsonville were celebrated with the Humane Society Award at the 2025 Regeneron International Science and Engineering Fair (ISAF), the largest global pre-college STEM competition. Their project, titled Studying the Effects of Fluid Shear Stress and Deformation on Metastatic Breast Cancer with Microfluidic Devices, reflects not only their individual dedication but also highlights the promising research ecosystem in South Carolina. The ISAF event saw nearly 1,700 students from over 60 countries presenting their research, making Howard and Zhang's achievement all the more remarkable. Their project was conducted during the GSSM Summer Program for Research Interns (SPRI) in collaboration with the Clemson University Honors College. Josh Witten, GSSM's Director of Research and Inquiry, expressed immense pride in their accomplishment, noting, "Their success not only reflects their hard work and scientific talent but also highlights the innovative research taking place in South Carolina and the importance of supporting student research at all levels of education." This recognition comes at a time when the scientific community is increasingly acknowledging the role of student-led research in addressing complex issues like cancer. Neville Erdrich, Vice President for Academics at GSSM, praised Howard and Zhang for their determination and curiosity which led them to present their findings on such a prestigious platform. Zhang reflected on the experience, stating, "We never expected it to lead to such an incredible experience. Presenting at ISEF was intense and awe-inspiring but also incredibly supportive. It gave me a glimpse into how bright and collaborative the future of science can be. In parallel with these academic achievements, the field of cancer research continues to evolve rapidly, with innovative approaches gaining traction. A new study has introduced an intriguing method for combating cancer by utilizing engineered fat cells to starve tumors of essential nutrients. This groundbreaking technique, known as adipose manipulation transplantation (AMT), leverages genetic modifications to transform white fat, known for energy storage, into beige fat, which burns energy instead. This reprogramming allows the fat cells to compete with and overpower cancer cells by consuming the nutrients that tumors rely on for growth. Dr. Nadav Ahada from UC San Francisco, who led the research, described the initial findings as surprisingly effective. In our very first transwell experiment, very few cancer cells survived. We thought we had messed something up. We were sure it was a mistake, he recalled, emphasizing the groundbreaking potential of this approach. The research, which has yielded positive results in lab dishes and preliminary trials in mice, signals a potential shift in cancer treatment paradigms, focusing on nutrient deprivation rather than direct cellular destruction. What makes this therapy particularly exciting is its ease of application in clinical settings, as the process of harvesting and reintroducing fat cells is already routine in plastic surgery. This opens doors for a new avenue of cancer treatment that could be both effective and less invasive than traditional methods such as chemotherapy or radiation. In another significant technological advancement, researchers at the University of Missouri have developed an AI tool capable of predicting the three-dimensional structures of chromosomes within individual cells. This innovation could revolutionize our understanding of gene functionality and disease mechanisms, particularly in cancer research. By enabling scientists to study the specific folding patterns of chromosomes, often varied even among cells from the same tissue, the tool provides a nuanced view of genetic activity that could lead to novel treatment strategies. These developments underscore a vibrant landscape of scientific exploration, with young minds and advanced technologies converging to tackle some of the most pressing health challenges of our time. As these promising projects unfold, they highlight a future where student innovation and cutting-edge research collaboratively pave the way toward breakthroughs in cancer treatment and beyond, offering hope and inspiration for generations to come. In a groundbreaking development for genetic research, scientists at the University of Missouri have unveiled an innovative artificial intelligence tool that can predict the three-dimensional structure of chromosomes at the single-cell level. This advancement has the potential to transform our understanding of how genes function and how certain diseases, particularly cancer, develop. Chromosomes, the compact structures that house our DNA, play a crucial role in gene expression. Each human cell contains approximately 6 feet of DNA meticulously folded into chromosomes to fit within the cell. This intricate folding not only conserves space but also regulates which genes are activated or silenced. However, when the DNA is improperly folded, it can lead to disruptions in cellular functions, which may contribute to the onset of serious health conditions, including various forms of cancer. Traditionally, researchers have relied on data derived from populations of cells, which averages the genetic information and masks the unique characteristics of individual cells. The new AI model, developed by Yanli Wang and Jianlin Jack Cheng from Mizzou's College of Engineering, addresses this limitation by providing a clearer and more detailed view of chromosomal structure within single cells. This is important because even cells from the same part of the body can have chromosomes folded in very different ways, explained Wang, the lead author of the study. That folding controls which genes are turned on or off. The challenge of studying single cells lies in the often messy and incomplete data they produce. The newly developed AI tool is adept at recognizing weak patterns in noisy data, allowing it to accurately estimate the 3D shape of chromosomes even when some information is missing. Remarkably, this tool demonstrates over twice the accuracy of previous deep learning methods when analyzing human single-cell data. In a bid to democratize access to this revolutionary technology, the researchers have made the software freely available to scientists globally. This open-access model enables researchers to leverage the tool for deeper insights into gene function and the mechanisms behind disease progression, potentially paving the way for more effective treatments. Our tool helps scientists study those differences in detail, which can lead to new insights into health and disease, remarked Chang, a Curator's Distinguished Professor of Electrical Engineering and Computer Science. Looking ahead, the research team plans to enhance the AI tool further by expanding its capabilities to reconstruct high-resolution structures of entire genomes. Their ultimate goal is to provide the clearest picture yet of the genetic blueprint contained within our cells. The initial findings of this cutting-edge research were recently published in the journal NAR Genomics and Bioinformatics. This development not only represents a significant step forward in the field of genetics but also signifies a broader trend in life sciences research where AI and automation are increasingly being integrated to enhance laboratory operations and data analysis. As these technologies continue to evolve, they promise to unlock new possibilities for understanding complex biological processes, driving innovation in treatment and diagnosis in the medical field. As we move forward, the implications of this research could resonate through various domains of health and disease management, ushering in a new era of precision medicine. In a remarkable convergence of technology and scientific innovation, researchers have recently made significant strides in nuclear fusion energy, drawing inspiration from the world of video games. This breakthrough, achieved by scientists at the Ulsan National Institute of Science and Technology UNIST in South Korea, promises to enhance the efficiency and stability of fusion reactors, potentially bringing humanity closer to realizing a sustainable, near-limitless source of energy. Nuclear fusion, the process of merging hydrogen atoms to form helium, mimics the reactions that power the sun. Unlike fission, which relies on splitting heavy atoms like uranium and generates radioactive waste, fusion offers the tantalizing prospect of clean energy without the environmental drawbacks. However, achieving practical fusion energy on Earth has long been hampered by high computational costs and the challenges of maintaining the extreme conditions necessary for fusion to occur. The breakthrough at UNIST involved the adaptation of collision detection algorithms originally developed for video games. These algorithms, which calculate the interactions of objects in a virtual space, allowed researchers to predict particle collisions in fusion reactions with unprecedented speed, up to 15 times faster than existing methods. As UNIST professor Isung Yoon remarked, the collision detection algorithm we developed serves as a crucial technological innovation. This advancement not only enhances the predictive capabilities of fusion reactors but also contributes to their overall safety and efficiency. The significance of this development cannot be overstated. In traditional fusion reactors, high-energy particles are injected to initiate fusion. However, these particles often collide with reactor walls, which can destabilize the reaction and reduce efficiency. By accurately predicting these collisions, engineers can design reactors that minimize unwanted interactions, thereby improving the chances of sustained fusion reactions. According to the researchers, the new algorithm reduces the calculations needed to predict these collisions by an astounding 99.9%, paving the way for more robust and operationally viable fusion energy solutions. The implications of these findings extend beyond mere efficiency gains. As the scientists noted, fusion energy has the potential to produce nearly 4 million times more energy per kilogram of fuel than fossil fuels, presenting a transformative opportunity for energy generation. The quest for practical fusion energy has seen its share of exaggerated claims and setbacks over the years. However, breakthroughs like the one from UNIST signal a tangible shift toward making fusion a reality. In parallel developments, scientists at the Wendelstein 7XW7X, the world's largest stellarator, have also made headway in fusion research by generating high-energy helium-3 ions for the first time. Utilizing ion cyclotron resonance heating, this milestone enhances the understanding of plasma behavior critical for sustaining fusion reactions. The resonance process that excites helium-3 in W7X could even shed light on phenomena occurring in the Sun, thereby linking fusion research with astrophysical insights. As researchers continue to refine their techniques and explore the potential of supercomputers to further improve fusion simulations, the future of this promising energy source looks increasingly bright. These advancements not only bring us closer to harnessing clean energy but also deepen our understanding of fundamental processes that govern the universe. The intersection of gaming technology and nuclear science may well lead to a new era of energy production, one where the clean, abundant power of the stars is within our grasp.